Hello and I welcome all of you to my channel Omni Bio Entrance. Today we will be discussing the National Entrance Screening Test, that is NEST 2019 Session 1. In this I will be discussing only the biology. Coming to the first question, arrange the following biological discoveries in the correct chronology from the earliest to the latest. First one, British biochemist Frederick Sanger introduces his plus and minus method for DNA sequencing. Second, American scientists Walter Gilbert and Alan Max introduced their method of DNA sequencing using chemical processes to terminate DNA strands. Third, Swiss physician Friedrich Miescher discovers and isolates DNA. 4. Canadian-American researchers Oswald Avery and Colin Munro MacLeod, along with American genetist Maclean McCarthy, working on experiments on pneumococcal bacteria, established that DNA could transform the properties of cells. American biologist James Watson and English physicist Francis Crick discovered the double helix when solving the three-dimensional structure of DNA, working from crystallographic data produced by Rosalind Franklin and Maurice Wilkins. So the chronology is, first is the third point, that is Swiss physician Friedrich Miescher discovers and isolates DNA. The fourth is by Avery, MacLeod and McCarthy for their experiments on pneumococcal bacteria establishing DNA could transform the properties of cells. Then the fifth one is by Watson and Crick discovery of the structure of DNA. And the first one will be the Frederick Sanger introduces his method of DNA sequencing. And the last one will be Gilbert Maxim method of DNA sequencing. So the correct option is option 1. Then each neuron has a cell body, axon and dendrites. The dendrites are the protoplasmic extension of the nerve cell. The axodendritic type of chemical synapse is formed by the association between the axon terminal of presynaptic neuron and dendrites of the postsynaptic neuron. So action potential causes the fusion of some synaptic vesicle to release the neurotransmitters for neurotransmission. Now this fusion of synaptic vesicle takes place in the membrane of the presynaptic neuron. Select the correct option from the following. Dendrites are nothing but extension of the axon terminals of presynaptic neuron. That's not correct. Dendrites are equipped with nodes of Ranvier. No, dendrites contain neurotransmitter-filled synaptic vesicle. They don't contain neurosynaptor-filled transmitter vesicle, and action potential stimulates fusion of these vesicle with its membrane. That is not true. Dendrites taking part in synapse formation are equipped with neurotransmitter receptors. Yes, they have neurotransmitter receptors but don't have neurotransmitter-filled synaptic vesicle to release the neurotransmitters for neurotransmission. So option 4 is correct. Then next one. The common morning glory Ipomia purpurea exhibits two flower colors where purple is dominant over pink. Within a population of common morning glory which is at genetic equilibrium, that is at Hardy-Weinberg principle, 9% of all the flowers are pink. Assuming that there are no other compounding factors, what is the frequency of flowers in the population that are heterozygous? So Hardy-Weinberg principle, they have given the frequency of the homozygous recessive one. So the purple is dominant and pink is recessive. So they have given the frequency of the recessive phenotype. So genotype. So that is Q square is equal to 9 by 100 and Q is equal to 0.3. We know that P plus Q is equal to 1. So P is equal to 1 minus Q, that is 0.7. According to Hardy-Weinberg formula, P square plus 2PQ plus Q square is equal to 1. They have asked the frequency of flowers in the population that are heterozygous. So we have to calculate 2PQ. That will give the frequency of heterozygous population. So 2 into 0.7 into 0.3, that is 0.42. Now next one. Seed traits of a given plant species following genetic inheritance are S long, small s short, capital W wrinkled, small w smooth, capital Y yellow, small y white, capital R ribbed, small r grooved. A cross was performed between two plants P and Q of the same species, wherein the plant P bears seeds that are homozygous recessive for grooved, homozygous dominant for long trait, and heterozygous for wrinkle texture and yellow color. The plant Q bears seeds that are homozygous recessive for short and smooth traits, homozygous dominant for yellow color and ribbed trait. Given that there are no influencing factors, state the resulting phenotype of the offspring from this cross. The answer is the seeds of half the plants would be long, wrinkled, yellow and ribbed. I have done the explanation in my next slide, so we'll go to that. But anyway, the answer is option. So they have given P plant which is homozygous recessive for grooved. So small r, small r. Then it is homozygous dominant for long trait, capital S, heterozygous for wrinkle texture, that is W, small w, and yellow color for that is heterozygous, so capital Y, small y. Whereas the Q plant is homozygous recessive for short and smooth traits, so small s, small w, and homozygous dominant for yellow color, capital Y, ribbed traits, capital R. So they have crossed these two plants, that is P and Q. So this is the genotype of the P plant and this is the genotype of the Q plant. These are the gametes that are produced. So upon crossing, we will get long wrinkled, yellow ribbed, long smooth, yellow ribbed. So half of the population from the offspring will be long wrinkled, yellow ribbed. So option D is correct. Then next one. Phylogenetic relationship among plants PQRS based on their shared characters are shown in the cladogram below. Identify the plant PQRS in the correct order. So we can see that vascular tissue develop in ferns. So Q must be ferns. Seeds develop in the gymnosperms. So R must be gymnosperms. And flowers are seen in angiosperms. So S must be angiosperms. So P must be liverworts. So option 2 will be correct. P liverworts, Q ferns, R pines and S lilies. Next one. Radioisotopes are frequently used in the study of cellular processes. A culture of E. coli is grown at 37 degrees Celsius in a minimal medium containing radioactive phosphorus. At the end of 48 hours, the amount of radioactivity expected in equimolar quantities of the following macromolecules. The macromolecules that they have given are RNA, enzyme and phospholipids. Now sulfur is a constituent of protein and hence enzyme will have only little phosphorus labeling in them. Then in RNA, phosphorus is present in more amounts than in phospholipids. So RNA will have more phosphorus labeling than phospholipids. So RNA greater than phospholipids which are greater than enzymes. So option 2 is correct. Now next one. The hemocytometer is a thickened glass slide with a small chamber of grids cut into the glass and is used to count cells. Each chamber has a fixed volume and is etched into 9 large squares. See figure P to X. Each measuring 1 millimeter into 1 millimeter into 0.1 millimeter deep. Each large corner squares P, R, V and X contain 16 small squares. With the cover slip in place, each of these small squares represent a volume of 0.1 millimeter cube. That is 1 millimeter square area into 0.1 millimeter depth or 10 raised to minus 4 centimeter cube. The number of cells counted in 4 large corner squares was 400 with the conversion factor as 1000. Number of cells per ml. Number of cells counted by large squares counted into dilution factor. Given this information, calculate the total number of cells suspended in a volume of 5 ml considering that the cells were diluted 1:2 before counting. I have done the explanation in the next slide. So first we have to calculate the dilution factor, that is total volume. How do we get that? Volume of the sample plus volume of the diluting liquid divided by volume of the sample will give you dilution factor. So you need 1:2 dilution, and you are asked to calculate for 5 ml of the sample. So half into 5 will be 2.5 ml sample, and the amount of diluent that you require to make up to 5 ml will be 2.5 ml. So dilution factor is equal to total volume divided by volume of the sample. So total volume is 2.5 plus 2.5 divided by volume of the sample, that will give you 2. Now they have given the formula: number of cells per ml is equal to number of cells counted by large square counted into dilution factor. So they have given number of cells counted as 400. So 400 divided by large square counted, that is 4, into 2, that is this dilution factor, into 1000, that is conversion factor, that is also given. You get 2 into 10 raised to 5. Now this is for 1 ml, that is number of cells per ml. You are asked to calculate for 5 ml. So 5 ml will be 2 into 10 raised to 5 into 5, that is 10 into 10 raised to 5, that is nothing but 1 into 10 raised to 6. So answer is 1 into 10 raised to 6. Next question. A scientist performed enzyme kinetics and obtained velocity v as a function of substrate concentration, and the values are given in the table below. The Km of the substrate could be. So the Km is the substrate concentration at which velocity is half the maximal velocity is the Km. Now if you plot a graph for this particular values of substrate concentration and velocity, you will get a typical Michaelis-Menten graph, and you can see that the substrate concentration at 800, the velocity values are not increasing, but they are going at a steady state. Over here, even if the substrate concentration is increasing, the velocity values are not varying, they are at a steady state. So this graph is also a typical Michaelis-Menten graph, and the Km is given by the substrate concentration at which velocity is half of Vmax. So this is the maximal velocity, that is 77.2, and half of it will be 38.6, and this is near that 38.6 value is near the substrate concentration of 200. So Km of the substrate will be 200. Next one. Which of the following statements are correct? The quantum yield of photosynthesis decreases with increasing temperature in C3 plants as compared to C4 plants. That's correct. The first stable product in C3 pathway is phosphoglycerate, whereas in C4 pathway it is oxaloacetate. That is also correct. C3 pathway is present only in C3 plants, and C4 pathway is present in all plants. No. The rate of photosynthesis is very high in C4 plants, very low in C3 plants. No. Answer is option 2, that is 1 and 2 are correct. The other 2 are wrong. Then, select the option containing the correct matched pair. Helper T cells. Helper T cells, they secrete cytokines as co-stimulators. So 1E. Then suppressor T cells. Suppressor T cells may downregulate or dampen parts of the immune response. So 2A. Phagocytes. Phagocytes ingest microbes or any foreign particulate matter, include neutrophils and macrophages. So option D is correct. Then memory T cells. Memory T cells are programmed to recognize the appearance of the original invading antigen. B cells differentiate into plasma cells that secrete specific antibodies. So option 3 is correct. 1E, 2A, 3D, 4B, 5C. Then next one. A small viral peptide can insert its 6 amino acids represented as a barrel. This is it. C. Into the lipid bilayer as depicted in the figure given below. So the barrel is inserted into the lipid bilayer. Now the table depicts the different properties of amino acids. They have given polar and uncharged amino acid, polar and charged amino acid, then non-polar and hydrophobic amino acids. The best suitable sequence that represents the barrel part. The barrel is in the lipid bilayer. So lipid bilayer is non-polar and hydrophobic. So non-polar and hydrophobic amino acid sequence will represent the barrel. So alanine, leucine, isoleucine, leucine, leucine, alanine, COH will be the sequence. Why? Because it is non-polar and hydrophobic amino acids. So option 2 is correct. Then next one. Proteins P1 and P2 bind to each other to form a complex. The complex has a high binding affinity to a specific element on the promoter sequence of the gene X. So P1, P2, they both combine and this complex has got a specific element on the promoter sequence of the gene X, thus transcribing it. The product of gene X is responsible for the accumulation of red pigment in some plants. So what is the product of gene X? It is red pigment coloration. Under drought stress, another protein P3 binds to P2 and prevents its binding to P1 and the plants appear pale. The following table depicts a representation of phenotype of wild type and mutant plants. So water sufficiency condition, that is the wild type plant will be red and in drought condition it will be pale. Now why is it red? Because the P1 and P2 complex, it bind to the promoter of the gene X and gene X is responsible for the red coloration. Now what happens under drought stress condition? There the protein P3 binds to P2. So P2 will not be able to form complex with P1 and then it will not be able to bind to the promoter sequence of this particular gene X. So gene X will not produce any protein products. So it will be pale colored. Now they have asked on mutant 3 has mutation in gene coding for protein P1 or protein P2. Now mutant 3, that is even in water sufficiency conditions, it should have given a red coloration, but it has given a pale coloration. So the mutant 3 has mutation in gene coding for protein 1 or protein 2. That is why even under water sufficiency conditions it is giving pale. Even in the drought condition also it is becoming pale. Because at water sufficiency conditions, the P1 and P2 should have bound to each other and it should have resulted in the formation of red pigment. But under drought also it is pale. So either P1 protein or P2 protein is mutated. That is why it is becoming pale in the water sufficiency conditions. Then next, mutant 2 has mutation in gene coding for either protein 1 or protein P3. Mutant 2. Mutant 2 is under water sufficiency condition, it is red. So P1 and P2 are normal there. Under drought condition also it is red. That means even if it is red at the drought condition, that means P1 and P2 can form a complex. So P3 is the one which is mutated because it is not able to bind to P2 and prevent the complex formation between P2 and P1. So the P3 should be mutated. What they have told is mutant 2 has mutation in gene coding for either protein P1 or protein P3. There won't be any mutation in protein coding for P1. So option 2 is wrong. Then mutant 2 has mutation in gene coding for P3. Now that is correct. Why? Because under drought condition it is showing red colors. That means P1 and P2 can form the complex. They can bind to the promoter of the gene X, thereby resulting in the red coloration. And P3 is mutated, so it will not be able to bind to P2 and prevent binding of P1 and P2. So option 3 is correct. Then mutant 1 has mutation in gene coding for P3. No, if it was having a gene coding for protein P3, then you would not have got pale colors under drought condition. So that is also wrong. Then following is the sequence of a gene that produces peptide P and has a promoter sequence in the 5' upstream region of the sense strand and not in that of the complementary strand. So 5' upstream region of the sense strand is having the promoter which produces the peptide P, but it is not present in the complementary strand. The promoter is not present in the complementary strand. Now if adenine is replaced with thymine and guanine is replaced with cytosine, so from this sequence, if we replace adenine with thymine and thymine with adenine, not just similar to how we are doing the complementary base pairing, adenine pairs with T, T pairs with A, G pairs with C, C pairs with D, it will get just the same way we will get a sequence after replacement. But here they are doing only the replacement and not altering the direction of the sequence in which it is aligned in both the DNA strand, then Chargaff's rule will not hold true in the case. Chargaff's rule will be true. Why? Because A is getting replaced by T and T will pair with A. T is getting replaced by A and A will pair with T. So Chargaff rule will hold true in this case. So the reading frame of the sequence is completely altered. No. Then Chargaff's rule will hold. Then transcribing complementary strand from 3' to 5' will not give the peptide P. Okay. The replaced complementary strand, after replacement, the replaced complementary strand will look the same as the sense strand, but in not the same direction, that is 5' to 3'. So complementary strand after replacement, you will get the strand replaced in the 3' to 5' direction only and not in the 5' to 3' direction. And they have told that at the 5' upstream region only the promoter for this particular peptide P is present. So even if the replaced complementary strand looks similar to the sense strand, the direction of the orientation of the sequences is not the same as that of the sense strand. Here in the sense strand it is 5' to 3', whereas in complementary strand it is 3' to 5'. So it will not give rise to the peptide P. So option 4 is also correct. Next one. DNA COT analysis is based upon the principle of DNA renaturation kinetics. The rate at which heat denatured DNA sequence in solution will renature is dependent on DNA concentration, reassociation temperature, cation concentration and viscosity, usually not a factor if the DNA is free of contaminants. Upon heat denaturation of an entire genomic DNA, the rate at which a particular sequence will reassociate in a given buffer solution is proportional to the number of times the sequence is found in the genome. Given enough time, nearly all of the DNA in a heat denatured sample will reassociate. Highly repetitive, moderately repetitive, and non-repetitive are the three types of DNA sequences found normally in all genomes, giving them different levels of complexity. Typical COT curves were obtained for genomic DNA from three organisms P, Q and R. The graph is in the next slide. So this is the graph. This is percentage of single-stranded DNA. This is on increasing size and this is the COT values. So genomic DNA of organism P is relatively less complex as compared to Q and R. Now, the shape of the COT curve for a given species is a function of two factors. One is the size complexity of the genome and the other is the amount of repetitive DNA within the genome. So larger the COT value, larger the complexity of the genome. So genomic DNA of organism P is relatively less complex as compared to Q and R. Yes, this is less complex. Q is moderately complex and R is highly complex. So P is relatively less complex than Q and R. That's correct. Then genomic DNA of organism R is relatively more complex than those of P and Q. Yes, R is relatively more complex than Q and P. So that is also correct. Next one. Genomic DNA of organism Q is relatively more complex than that of R. That is not correct. Genomic DNA of organism P is relatively more complex than that of R. That is also wrong, because larger the COT value, the more complex is the genome. So option 1 and 2 are correct. Next one. Consider the plant species. P is more efficient than plant species Q in nutrient uptake. Plant Q is better seed disperser. So P is more efficient than Q in nutrient uptake, but plant Q has got another positive quality, that is, it is better seed disperser than P. In this example, the resource under competition is nutrient. So competition for nutrient is the example that they have chosen. But nutrient acquisition is related to availability. Okay. If a disturbance opens up new space for colonization, plant Q is expected to arrive first. Why? Because it is a better seed disperser. So it will arrive first at the new space and maintain its presence in the community until plant P arrives. And it will be the king in that particular new space even before the plant P or until the plant P arrives. Why? Because it is a better seed disperser and begins competing with plant Q. So it will be the king in that new place until plant P arrives and start competing with Q. Eventually, plant P will outcompete Q because plant P is more efficient in nutrient acquisition and grows faster. What happens when P arrives is that the plant P will outcompete plant Q, and because it is more efficient in nutrient uptake, and it will grow faster. With an increasing plant P population, the plant Q population will decline, and given enough time, has a possibility of shrinking in the number in the present habitat. So in the new habitat, plant P will be more efficient nutrient uptaker, and it will increase in population, and it will result in the decline of growth of the population Q. It can come to such an extent that shrinking the number of Q in the present habitat will take place. The ways of preventing the declining numbers of plant Q could be. So how we can prevent the plant Q from declining its numbers? So intermediate local disturbances such as uncontrolled harvesting of plant P for commercial gains. So that is one option. If you start harvesting plant P, the number of plant P will start getting a bit reduced, and this will favor the growth of plant Q. Then sudden and specific infection of plant P by a fungus. So if there is a sudden and a specific infection of plant P, that is, specific infection means this fungus is not going to infect plant Q, then there will be decrease in the population of plant P by the fungal infection, and Q will be able to survive, and it will not go up to the level of shrinking. Then improved seed dispersal strategies by plant P in successive generation of plants. If you improve the seed dispersal of plant P, which is already competing with plant Q and leading to the shrinking population size of plant Q, so improved seed strategies for the plant P in the successive generation will again help in P to outcompete Q. Then producing compounds which inhibit the growth of plant Q. Now already plant Q is lesser in population, and if you produce compounds that inhibit growth of plant Q, naturally you will be going to reduce or make the plant Q extinct in that particular area. But they have asked for the ways of preventing declining numbers of plant Q could be. So this is the option 1 and 2 are the remedies for that. So thank you. Please like, share and